Hello everyone, thank you all for joining us today. This is the webinar from Alvéole. I am Luc Talini, CEO of Alvéole, and I have two guests today with me, Pierre-Olivier and Vincent. Perhaps Vincent, you can introduce yourself.
Hi Luc, hi everyone. I'm Vincent Studer and I'm a researcher at the CNRS, more precisely at the Interdisciplinary Institute for Neuroscience in Bordeaux. And I'm in the lab where the Primo technology and the Primo has been developed.
Thank you, Vincent. Pierre-Olivier.
So first, thank you, Luc, for this nice invitation. So my name is Pierre-Olivier Strale. I'm working with Vincent in Bordeaux as a postdoc, and I'm a cell biologist and a bioengineer.
Okay, thank you. So this is Alvéole's first webinar, and we are very pleased to present you Primo, what you can see here on the image, the first platform for multi-protein patterning. We are going to spend about 45 minutes together to talk about the technology and its application in cell biology. But before starting the webinar, I want to go over some technical details. First one is you can interact with us thanks to the chat box that you certainly can see on your windows. Please feel free to ask any question that you have about the technology and its application. And I think we're going to have time at the end of the webinar to answer to your questions. And this webinar is being recorded. That means that we will be sending you a link by email to be able to see the video, I think, in 2 days. And we also post the address of the video on our website in 2 days. Okay, so now we can start the webinar. So we are going to talk about this beautiful white and green box that you can see on the picture, the Primo device. And when you combine this device to the reagent that you can see on the picture, you can print proteins through the pattern that you have decided to print, as you can see on the image. And just to see how to use the device, if I can change the... Yes. So here you can see the setup. So the box, the green box is directly docked to a microscope. Today the best configuration is obtained with Nikon TI. And when you combine this box with the reagent, you can pattern, photopattern such a figure that you can see here, a beautiful yin-yang pattern with two different proteins. And in this experiment, it's used for a co-culture experiment. So now we're gonna explain how it works. So Vincent, sorry, this is this one. So can you say a little word about the technology?
Sure, Luc. So I'm going to try to explain in a few words how the technology that we developed works. So the technology we developed is called Liap for light-induced molecular absorption of protein. And as for many protein patterning techniques, it starts with a substrate which is covered with an anti-fouling layer of polymers. So in our case, we use polyethylene glycol. So a brush of polyethylene glycol is deposited on a glass cover slip. And then we add a reagent that we call PLPP. And this reagent is photosensitive. It means that when it's illuminated with UV light, that photoinitiator is activated and can locally degrade, by a photoscission mechanism, it can degrade the polyethylene glycol. So it means that wherever you illuminate your substrate in the presence of the PLPP, you remove the PEG, the anti-fouling PEG coating. Then, as you can see on the fourth sketch, if you incubate a protein, it is now able to adsorb wherever the PEG has been removed. So in a word, how do we project our pattern of UV light? We use a system that is very similar to a video projector, and this system is what's inside the Primo apparatus, except that it's a little bit more complicated than a standard video projector. It is based on a UV laser that is shined on the DMD, which is a chip composed of individual micromirrors that you can switch on and off to create an image. And then by an optical relay that the image of the DMD is projected onto the glass cover slip at the micron scale. And very interestingly, this system projects the light on the entire field of view, so it's a whole field image illumination process. And the other interesting thing is that you can make grayscale patterns of light with that system. So now what we're going to see in a video is the Liap process in real time. So what we did here is to illuminate in presence of PLPP, a fluorescent protein, namely GFP. And as soon as we shine light on this system, the protein is able to adsorb on the substrate with a given pattern. So if I send the video, you're going to see in real time the GFP molecules adsorbing on the surface wherever it has been illuminated. So as you can see, it's a very fast process. So not only it is fast, it is also quantitative, which is a great advantage of the Liap technique. For instance, if you submit a pattern like you can see on the A image, composed of squares of different intensities, what you end up with is a pattern with different densities of protein adsorbed on the surface. And what you can see on the curve on the image C is that you have an almost linear dependence of the protein density with the dose of UV that you send. With this, you can indeed create a very complex gradients of protein. And then the other aspect of the Primo system is that it has been well optimized to have a very good resolution. So as you can see on the image A, we can end up with patterns with a resolution which is close to the diffraction limit. Here, lines separated by only 500 nanometer. And on the right, 2 micron dots separated by a few microns. Interestingly, we said it in our introduction, Primo is a multi-protein patterning method. It means that the process that I just described can be repeated many times. So if you want, you can introduce for a second time, you create a first pattern, like I just described, and then you can put PLPP again, and if you send another pattern of light, and incubate a second protein, you can make patterns composed of multiple proteins. As you can see on the bottom right, an image of a pattern composed of 2 proteins, GFP in green and M-cherry in red. And what you can see also is that on the outer square, the 2 proteins are present, means that you can have either one or the other or the 2 proteins. So to really demonstrate the performances of the Primo and Liap, we tried to reproduce at the micron scale a famous painting. And to do so, you only have to separate the image in its 3 different color components, namely the red, the green, and the blue component. And at the same location of your substrate, you can you can pattern 3 different proteins with 3 different colors. And if you do this, and then take a fluorescence image, you end up with a micron scale reproduction, which is very nice, as you can see, of this painting, composed of 3 biomolecules with 3 different fluorescent tags. Of course, you can not only do nice paintings, but for us, the Primo and Liap, we use it in our lab to make biological experiments. And in our case, we're interested in doing gradients of proteins with a controlled shape. So here, for instance, on the D picture, we try to make gradients with either a linear, an exponential, or a logarithmic shape. And as you can see on the graph E, the pattern we send to the apparatus with our computer in blue matches pretty well the pattern we obtain eventually. And of course, again, you can make cross gradients composed of 2 opposite gradients composed of 2 molecules, here in red and green, as you can see in F. So really, Primo is a quantitative method allowing to do multi-protein gradients. Oh, sorry, I think I went 1 slide too far. So in those examples, I described how to pattern proteins, so for instance, purified proteins directly, but there are other strategies that you can use to graft proteins. So if you want to graft your proteins in a given orientation, for instance, you can use antibodies for tags. So what I call immunobinding of tag proteins. So you can use anti-FC, anti-HA antibodies, for instance, that you pattern using the Liap technique. And then you incubate your protein of choice. this protein has been previously tagged with the corresponding tag FC, HA, and so on. So if you do this, you can orient your protein. You can also use the standard biotin streptavidin link. So you can, for instance, pattern biotin on your substrate, streptavidin on your substrate using Liap, and then incubate your biotin-related protein. Finally, I would like to mention that you can also do what we call inverse photopatterning, for instance, by using functionalized PEG. So if you start with a brush of biotin-related PEG, for instance, using Liap, you can locally remove those biotin-related PEGs. Then you can incubate a protein, for instance, which is directly going to where you have removed the PEG. But now, if you incubate, for instance, streptavidin and then a biotinated protein, it's going to go on the outer part of the pattern, which we call inverse photopatterning. So with all those strategies, you really can find, for almost any given protein, you can find a way of grafting it to your pattern in a given shape and with a very good resolution.
Okay. So thank you, Vincent. I think we have seen very well how we can use this technology, and now I would like to talk a little bit about the application of protein printing, and perhaps Pierre-Olivier, you can tell us more about what we can do with Primo and the protein printing.
Sure. So as Vincent told us just before, Primo is a highly flexible technology and allows for generating patterns of any shape and any size. So in the example I'm going to talk about right now, rectangular patterns of fibronectin were printed and fibroblasts were allowed to adhere on these patterns. So first, as you can see, the cells adopt a rectangular shape. This is the shape of the pattern. The pattern occupancy is very high, is very close to 100%. And very interestingly, the anti-fouling properties of the substrate last at least 2 weeks, making Primo a suitable tool for long-term time-lapse imaging. So in this second example, patterns corresponding to the letters making the word alvéole were sequentially projected through a 10x objective, and after addition of fibronectin, fibroblasts once again were seeded. So after washing away the poorly adhesive cells, the presence of the pattern is revealed by the presence of the cells. Once again, the pattern occupancy is very high, almost perfect, and the non-specific adhesion of cells outside of the pattern is very low. So altogether, Primo allows for fast and specific printing of multiple cells in a highly flexible manner. So for the next two examples, the multi-protein printing capabilities of the Primo technology was used in order to first control dynamically the behavior of a single cell. So concretely, a first pattern of series 3 label fibronectin was performed, corresponding to the right part of the yin-yang, and that's what you can see in the C panel. Then a second pattern composed of 647 streptavidin was done, and it corresponds to the left part of the pattern. Then mouse embryonic fibroblast cells were seeded, and in this experimental context, the cell can only adhere to the streptavidin pattern, to the fibronectin pattern, sorry, the right part, the green part of the yin-yang. And at a given time point, a solution of biotin-related fibronectin can be added, and thanks to the biotin-streptavidin interaction, this biotin-related fibronectin will attach to the left part of the yin-yang, and this allows the cell for spreading on the full yin-yang circle over homogeneous non-adhesive background. So a very similar protocol was followed in order to perform co-culture experiments with an exquisite control over the cell type's localization. So a first pattern corresponding to the right part of the yin-yang was projected through a 10x objective, and fibronectin molecules were allowed to adhere on such pattern. A second pattern corresponding to the left part of the yin-yang was subsequently performed, and streptavidin molecules were allowed to adsorb. At this moment, a first population of cells, so in this case we used S180 cells expressing icadherin GFP cells, are seeded, and these cells can only attach to the right part of the yin-yang composed of fibronectin. After addition of a biotinylated fibronectin solution, exactly as I mentioned before, you can wash away the excess of this solution and seed a second cell type. In this case, we use mouse embryonic fibroblast. And as you can see, the two different populations of cells are properly localized on their given pattern. So altogether, the examples I showed you just before illustrate how efficient the Primo technology is in order to control cell positioning, cell behavior, and also in order to generate a very complex co-culture experiments. Okay, so with the help of our collaborator, Sonia Garel and Ludmila Lockman at the École Normale Supérieure in Paris, we recently designed an experiment in order to better understand the axonal guidance in response to chemical cues. So what you can see on the top left panels are thalamocortical neurons migrating through a corridor, and this corridor is highly enriched in guidance cues. So the two populations, the green one and the red one, originate from the same area of the brain, but they migrate in very distinct regions of the cortex, maybe because of local gradients of guidance molecules. So we aim at understanding the attractive or the repulsive effect of immobilized guidance molecules. And to do so, we pattern gradients of molecules at the end of a triangular pattern, and that's what you can see on the bottom left panel. Then a brain explant is put at the tip of the triangle, and after 2 to 3 days in culture, the axon starts to migrate and finally reach the gradient area, and eventually they respond to the local density of guidance cues. So thanks to its quantitative printing capabilities, Primo is a promising tool for those who need to finely tune the local density of adsorbed biomolecules.
Okay, thank you. Now we are going to talk about something else, because in fact, as we have a great technology for UV photopatterning, we can use this technology for other application. And here I would like to discuss with Vincent about the way to manufacture hydrogel with Primo.
Indeed, Luc, so far we saw how to use Primo for protein printing, but there's many other things you can do with this machine. If you understood well, Primo is in fact a wide field projection apparatus for UV light, namely 365 nanometer wavelengths. And there's many photolithographic or photopolymerization processes that are active at these wavelengths. So you can use Primo, for instance, to generate photopolymerized hydrogels. So, for instance, if you want to make hydrogels made out of PEG acrylate in water, so in our case we use 95% of water with PEG acrylate, what you can do is use, for instance, a small microwell. In our case, we make it with PDMS, and I think you're going to see that later in the movies. But what you can do is project a pattern of UV light in this mixture of PEG acrylate and on photoinitiator, PLPP. And if you do that, as you can see on this movie, we very quickly photopolymerize the logo of Alvéole in a PEG hydrogel. And what you can see also is that compared to other methods, it's very fast, because the optical power, the intensity of light that we have with Primo is quite high, so you can very quickly photopolymerize hydrogels. And then, in the same way that we made multicolor patterns of protein, multi-protein patterns, you can also make a multi-hydrogel patterning, that is, use different types of hydrogels and combine them nicely to make a microstructure. So in this example on the bottom of the slide, we made a flower composed of 3 different types of hydrogels. One is colored with FETC, the other one with 3TC, and the third one is biotinylated hydrogel. And then if you incubate fluorescent quantum dots coupled to streptavidin, they nicely only coupled to the circle hydrogel in the center, which contains the biotin. So this shows nicely that you can make multi-functionalized hydrogels with this method.
Okay. So, and perhaps we can go further with this technology, because as you mentioned, we can use many material sensitive to UV. And of course, we can also use Primo for micro-manufacturing. And can you tell me more about that?
Exactly. Many people actually are using microfabrication in their labs, for instance, for doing microfluidic channels or PDMS microchips. So the first step for this type of application is usually to make a photolithographic mold made out of photoresist. And usually you use apparatus made for microelectronics, but you can also use Primo to make molds made out of photoresist. So you start in a similar way as for other techniques. You use a silicon wafer that you cover with your favorite photoresist, and then with your computer on Primo, you can send this pattern of light onto your silicon wafer and make really nice lithography. So here's an example of a mold made out of SU8, which is a very commonly used photoresist. This mold is composed of little micro square, which are 190 micrometer wide. And as you can see, we made a nice lattice of those micro wells with Primo, with a very nice definition. I mentioned the microfluidics. You can then use such types of molds made out of photoresist to manufacture microfluidic channels. So this is what we did. We did a microfluidic channels made out of PDMS, molded on a SU8 photoresist master, fabricated with Primo. And then, again, you can use Primo to now, for instance, polymerize hydrogels inside that PDMS microfluidic channel. So this is what we did in the geometry that you can see on the left. And now I'm going to show you a very short movie showing how we photopolymerized an hydrogel at a given location of a microfluidic channels. Because due to the fact that you can control easily the shape and the localization of your illumination, it's very easy to do such experiment. So as you can see very quickly, you can see that rectangle, which is a photopolymerized hydrogel inside a microfluidic channel.
Okay, so thank you, Vincent. Thank you, Pierre-Olivier. We have seen many things that we can do with Primo. Now we have prepared a series of videos to see how really we can use the technology and the different steps for obtaining the protein printings. So if you can start the video. So here we can see the installation of the device. So it's very easy to install at the back of the microscope. Again, here the best configuration is Nikon TI. And so now we have now the first step, the chemistry, the surface chemistry. You need to treat your glass slide in order to obtain an anti-fouling coating. So can you say a word about that, Pierre-Olivier?
Yeah, of course. So we use a clean glass cover slip. And the first step consists on putting this glass cover slip in a UV plasma chamber so that you can activate your surface. So it takes around 5 minutes.
And if you don't have a plasma cleaner, we can provide the user with pre-coated slides also. So why a plasma cleaner?
The idea is to activate the surface so that you can properly adsorb your PLL peg just after. So now you can see that the plasma treatment is done.
And so the next step is to coat the slide with the PG. So can you say a little word about what we are seeing on the
Sure, yeah, these are the PDMS microwells that I mentioned before and that we use to properly put our reagents on the cover slip. So these microwells are actually made in a thin foil of PDMS that is about 200 micron thick, and that we can cut with a vinyl craft cutter. And once you put that foil of PDMS on top of your cover slip, you're going to see that you can nicely put very small volumes, in this case of PLL PEG solution. With only a few microliters of solution, you can cover millimeter square of your cover slip.
Here it's a glass slide, but I know that we can use this technology with other material also.
You're right. Yeah, we tried and we successfully tried to pattern different types of substrates, such as plastic petri dishes, PDMS, and so on. You can even do this in multi-well plates or things like this.
And here we can see the pre-coated slides that we can provide. So you have to wait, I think, 1 hour.
Yes, in standard time for PLL PEG incubation is about 1 hour. But there's different processes to put pegs on cover slips. On some of them, for some of them, you can actually store the cover slip with pre-coated cover slip.
So yeah, what are we watching?
So what you can see now, Aurélien is washing away the excess of PLL PEG with physiological solutions such as PBS. And we're gonna see maybe in the next movie now.
Yeah, because now we are ready to print.
Yeah, so now
proteins.
the cover slip is ready for printing. But I think now what we're looking at is the calibration procedure. Because indeed, in order to have the pattern at the right size and at the right position, there's a calibration step which we make with a cover slip, which is only coated with a thin layer of Stabilo Boss fluorescent marker.
And yes, it's a function of the software.
True. So the software sends a dedicated pattern, namely 4 squares, and then the camera on your microscope is taking a picture of that fluorescent pattern, and the software can automatically generate a calibration so that the pattern you send nicely match your camera image.
And just a detail, as you can see, the software is in French because we are a French company, but it also exists in English, of course. Perhaps the next video with the printing.
Yeah, so now the software is calibrated, and I think now we're ready for to make a protein patterning experiment. So the first thing is to remove the PBS or the buffer that has been left in the microwells. So that's what we're doing now. And then you can add the PLPP, so the photo initiator. So only a few microliters of PLPP are necessary to cover the surface of the microwell, as you can see. And then what you can do is just select your pattern of choice with the software.
And how do you draw your pattern?
So that's the good thing about the Primo, is that you can use any type of TIFF or kind of image, grayscale or binary image that you can load in the computer. So you can use any drawing software to make your patterns.
With a standard format?
True, JPEG, TIFF, any image. So here what you see is the UV light that is projected. So the pattern is projected. And now...
How long?
It's only a few seconds. It was almost real time. So now it's time to remove the PLPP and to add, I think, the protein.
Yes.
So
How long? Yeah, it's written 5 minutes.
So it depends on the concentration of the protein, but usually it's a few minutes.
Which concentration, Pierre-Olivier?
It depends. You can, depending on the size of the pattern you want to use, but usually around tens of microgram per
so standard
milliliter is okay. Yeah.
Okay.
So we almost didn't see, but it was quick. But we saw the result of the patterning.
Okay. Perhaps the next video about all the possibilities
okay, so
of the software.
So in this case, I think we're going to see how to pattern multi-patterns. So it's usually it's very useful to be able to reproduce the same pattern many times. For instance, if you want to reproduce your experiments multiple times. So in this case, I think the software is configured to make a 2 by 3 array of the same pattern. Here you can see that you can control the dose of UV that you're going to send. And then the software combined with the motorized stage and followed by the protein absorption protocol, you can generate many copies of that grayscale patterns, as you can see.
So here we are seeing the patterns with the camera.
True, yeah. The software is scanning through the different patterns, grayscale patterns.
Okay. And so here we have...
So I think we didn't see the size, but I think this pattern was made probably with a 20x lens. So again, the width of the pattern is a few, maybe 100 or 200 microns.
Okay, so I think we have another video for the other possibilities with Primo and the software. Okay, so yeah, here we have things. It's not protein printing. Perhaps you can say a word about that.
Yeah, that illustrates what we mentioned previously. That is the fact that you can use Primo to photopolymerize or photopattern other materials. Here in this case, it's photocurable glue, namely NOA 81. And again, you can use this polymer to make microstructures. And here we illustrate an interesting feature of the software, which is when you want to make patterns that are larger than your field of view, maybe millimeter-size patterns, such as the microfluidic channel I showed before, you can use the stitching procedure of the software, which is going to separate your large pattern in a different sub-pattern, and combine with the stage of the microscope, you can create a nice millimeter-scale pattern at the micron scale.
Okay, so I think that was the last video about, no, sorry, another function of the software. There are many functions in the software. The possibility to align the pattern with another pattern or with a device.
Yeah, because for instance, in our lab, we saw that it's important that many times we have to go back and forth between the cell culture room and the patterning experiments. For instance, if you want to seed cells, and what's interesting is that you can recover the alignment of your sample between those steps. And in this case, the software is able to recognize a first pattern that you've made. You can then align your software to that first pattern. And if you make a second pattern, in this case, the A of the logo Alvéole,
and the accent,
you can nicely, on the accent, true, you can nicely align it to the first pattern that you made. So
it's very precise.
Yeah. That alignment is again at the micron scale.
Okay, so a very useful function for multi-protein patterning. So okay, so I think we have finished with the video, and I think we have some questions of the people who are looking. Yes, no, first, sorry. We are going to talk about how to obtain a Primo. So perhaps you can see. So the offer. So. First things is for a Primo you need a setup. As I mentioned it today, the best configuration is a microscope from Nikon, the TI from Nikon. You also need a motorized stage if you want to make multi-pattern or big picture, and also camera if you want to check your pattern. And now in Primo you have several things. First, the box, of course. In this box you have a UV laser and a video projector, a DMD. We also offer 1 year of reagent. It corresponds to 3 milliliter of PLPP, and with that you can make about 500 printing. You have the software. We have described the different function of the software, Leonardo, and 2 days of for installation and training on site. And we have a premium offer, which is available till the end of June, and the price is 65,000 euro for the complete offer. So now I think we have time to answer to the question we have from the people who are watching us. So I have a first question. I don't know who can answer to this question. I use a lot of 96 and 384 well plates. It is possible to consistently print the same pattern on many wells of a multi-well plate. So,
yeah, I think I hope that we showed what we showed. Partially at least answers the question. Indeed, it is possible to pattern inside multi-well plates, even in plastic multi-well plates, because you can print on polystyrene substrates. And again, the software is able to do a multi-patterning with a given pitch, so you can adapt the pitch of the software to any type of multi-well plate. And the only step that for now has to be done by hand is the rinsing steps, but maybe if you have a rinsing robot or something like this, you can very easily make patterns, different patterns in your wells.
Okay, thank you. I have another question. Is there other techniques for patterning proteins? What are the advantages of your technology? Vincent again.
Again,
I think we have a slide about that.
Yeah, because we're not the only one to have developed a method for protein patterning. Many people, for instance, have used micro contact printing for protein patterning. So maybe it's since we have this slide prepared, we can I can say a word about the different techniques. So microcontact printing is a very commonly used method based on silicon stumps made out of PDMS. And the idea is again to start with a cover slip that is covered with an anti-fouling layer, for instance, of polyethylene glycol. And then what you do is you ink your stamp with your favorite protein, and then you put that inked stamp on your substrate, and you're going to deposit the protein at the given location. So it's a very cheap and easy to handle method. I would say that there's a few drawbacks. It's hard to make multi-protein patterns because you would have to align your stamps between the different steps. And the other thing is that the density, the density of protein is the same on the entire stamp. So there's, to my knowledge, there's no way to make gradients of proteins with this technique. Then there are other techniques based on light, so namely laser patterning. So the idea of laser patterning is to use, for instance, a photoresist that you can remove with a scanned laser. So this technique is, in a sense, similar to Liap, except that it's a scanning technique. So it's very slow because you have to scan your laser spot on your entire cover slip. So to make let's say a few millimeter squares of patterns, that would take a very long time compared to Primo. And the third method is photopatterning made by photolithography. So in this case, you use chromium masks on quartz, which are commonly used in microelectronics. If you do this, you can also use a photoresist, remove that photoresist locally with where you shine light. Again, this technique is limited to, I think, to maybe only one protein, because if you want to make multi-protein, then you would have to align your mask for the different proteins, and it's very cumbersome. So, in one word, I would say that we try to improve and take the best out of those three methods when we developed Liap, because I think Liap is really the only method which can make multi-protein quantitative to make gradients of multi-protein and fast. That's important.
Okay, thank you, Vincent. Perhaps a question for Pierre-Olivier. We have partially answered to this question, I think, but does it work with other material like plastic of culture dishes?
Yeah, exactly. So we already answered this question before, but we have been able to pattern proteins in a quantitative manner on glass, cover slip, on PDMS, and also on plastic petri dishes such as the multi-well plates Vincent mentioned just before.
Do you have to change the chemistry?
No, the chemistry is rather similar. The only one thing you have to adapt is UV dose, because UV dose you have to project is somehow different between the plastic and the PDMS, for example. So
okay, so you have to tune.
You have to tune your illumination
okay,
so that you can pattern your protein.
Okay, thank you. A third question: will it be possible to attend to co-culture cells on two different matrix proteins like this without biotinylated one protein? For example, could I make the yin-yang with the yin laminin and the yang fibronectin?
Okay, so we haven't tried that yet, but I don't see why we couldn't do that.
Okay, yeah, because you have already print all this of protein separately. Laminin, I think it's okay, and the fibronectin also.
Yeah, I think if you have a good set of cells with the corresponding matrix, there's no reason why you could not pattern both of them in an intricate pattern.
Okay. Perhaps the last question, perhaps more for me. No. Is it possible to try the system before ordering it? Yes, it's possible. You just have to send us a mail and we can discuss that, because we really want to see Primo in different labs worldwide. So do not hesitate to contact us and to see if we can make a collaboration for land. I check the questions. Okay. 3 most important typical problems in lab that Primo will address. Difficult. The best 3.
Okay, so I would say, as Vincent mentioned just before, I think this is the only one technology that allows for printing of gradients of molecules in a highly flexible manner and very rapidly. So I think, as example I mentioned before, the experiments for guidance molecule, I think that the kind of experiment you cannot do without the Primo technology.
So yes,
So this
gradient one.
Gradient
And
is
two?
one. Of course, Vincent.
Well, another one I think is what I described about hydrogels and photopolymization. I think labs which are not equipped with a clean room facility with Primo, they could in their for instance biology lab have access to these very useful technologies with only one equipment, and I think that's really
An open
asset.
instrument, for
Yeah.
many applications, and for free if we have.
The last one, I would say that the only one apparatus that allows for doing some very well-controlled co-culture experiments. There is no way to control precisely the positioning of different cell types so far. So I would say that the third
we
advantage.
really hope that there's going to be applications, for instance, in tissue engineering with this type of technique. So we're not expert in that, but I really hope that labs which will have this Primo in their hands will use it for such tissue engineering applications with very well-controlled localizations of different cell types.
Yeah, because the resolution is 1 micrometer.
So the resolution is much better than the size of the cells. So you can really control the location of the cell at the subcellular scale.
Okay, thank you. I think that was the last questions. So thank you all for watching this webinar. Perhaps the last word, Alvéole will be attending in a meeting in Barcelona, the Advance in Cell and Tissue Culture from the 13th of May to the 1st of June. We're going to have a booth there, and so if you are around, we will be very pleased to meet you directly at our booth, so do not hesitate to come to see us. So thank you again, Vincent. Thank you,
You're welcome.
PO, and thank you all, and I hope to hear about you soon. Au revoir.